I'm very interested in the precision medicine approach that your company represents and how we can potentially test to see, how this effectiveness and how precise it is and what we're able to do with it, I think is obviously transformational and definitely something that needs to be done as a high priority.
We've been investigating the opportunity for using existing drugs that are safe and well tolerated. What do you think the possibilities are for drug repurposing? Is this the way forward for these diseases? Welcome to Biology Matters, the podcast exploring how a deeper understanding of biology can transform healthcare at a time when it's becoming globally unsustainable. I'm your host, Steve Gardner. Let's get into it. So I'm joined today by Chris Armstrong, who is the director of Melbourne ME CFS collaboration and is a scientific advisor to the Open Medicine Foundation. It's a delight to speak to you on the podcast, Chris. I am very grateful that you're joining us. You're taking time out of family vacation to be with us. But thank you very much for that.
Yeah, no problem at all. I think I very much appreciate getting on here. I think we've discussed me coming on for a little bit, and I'm glad that we could make some time now while the little ones are having a bath effectively here in Melbourne. I know it's pretty early in the morning there, so I appreciate you making time for me there too.
Oh, no problem at all. No problem at all. So, Chris, I mean, these days you're at the Bio21 Molecular Science and Biotech Institute in Melbourne. I realise that I've only really known you in that role, and I don't know much about your early career. Where did you get started, and how did you get into MECFS?
So I did my honours. So honours degree. So I did a Bachelor of Science at the University of Melbourne, and then I went on to do an honours degree. And an honours degree, or an honours portion of this actual degree, is allowed or enabled in Australia. It's almost like a fast masters for people who want to go or know that they want to go and do a PhD.
Yeah.
So it's a full research year, and then after that, you are eligible to make it towards a PhD within Australia. And so when I actually joined up, it was to learn metabolomics, and metabolomics, because I was very interested in this new field at the time, the idea of measuring lots of little small molecules that exist in biofluids within people that can potentially serve as markers or biomarkers of their disease. And it was being utilized in, diabetes and obesity and cancer and a number of different conditions. And we had some cool tech and cool pieces of infrastructure at the University of Melbourne called NMR, which are nuclear magnetic resonance spectrometers, which are very, very big pieces of machine, large magnets. And they've largely been utilized to look at proteins. And it was a protein lab. And they were actually interested in recruiting me on to do an honours project to produce or to bring in and develop the metabolomics pipeline for those machines. And so I did that as part of an honours project in the context of looking at a few patients with MECFS. And that was my honours year introduction. And I think working with the patients and looking into the field, and even back then, which was back in early 2010, back then I could feel, you could see that there was kind of this emerging evidence of a biological problem, while it was still in this interface and still trying to get people to believe it as a biological problem. And so I found that quite fascinating, because even from my project, I found some interesting biological differences. And so I thought, well, I better go on and continue to do this for a PhD. And that's how I really got into MECFS. But running clinical trials, you get to meet patients, you get to meet kind of human stories with this associated with this illness, and that I think draws people in. I see that now even with students in our group who are effectively young and being kind of that's face-to-face with patients, and it does draw them in, definitely.
I think that's one of the oddities of a number of diseases that were studied. They're not necessarily very well understood by the general population, and yet they are incredibly debilitating for patients living with those conditions. Has that given any particular challenges either in getting studies funded or in telling the story of why work in ME/CFS and other related diseases is so important?
Yeah, absolutely. I would say, look, it's always been difficult to get funding within this field. It was
Mhm.
increasingly more difficult, I would say, even back then, um, because it takes a lot to get recognized. I think through the large government-based funding, peer review process within academia extremely important, because and a true way to kind of keep things fair and keep science progressing. Um, but one of the main limitations of it is that it relies upon viewing researchers based on their track record within a disease
Mm.
to give them notoriety or to make it seem like they kind of know what they're doing within a space. And so it's actually very hard to crack that egg and crack into that. That kind of creates this kind of club that you're not a part of when you start and get in there. Because it's like, how do you show that you're credible when you haven't ever had the funding to do a large study? And even if you show, a sequence of small studies, how do you show that you can run a large study, that you deserve, you know, hundreds of thousands of dollars of government money to do a research project? And that's where it's been extremely difficult. And that's where, philanthropic bodies coming in has been kind of a lifesaver and actually triggered kind of funding within this space, I think, at a small level for a long time to get a community growing. And I think, they've done a great job in what they've been able to do.
I think from that PhD, from your initial project, possibly coming out of your PhD, you ended up publishing one of the very first papers on metabolomics in ME/CFS. How was that received? And you talk about having to break the door down a little bit in terms of credibility or notoriety. Was that immediately accepted? Was it something that fitted into the canon of explanations of the disease biology?
It was well received within the research community. And actually, for the first many years, actually during my PhD, I largely existed within the conferences of metabolomics. For me,
Right.
making sure the methodology and stuff that we were doing was the main focus at the time. And it was really at the end of my PhD that I would go to conferences, and I didn't realize how many people had kind of been reading that work and been wanting to talk about it. So it was very well received within the research community, from what I could tell. And at the time, there was quite a bit of a divergence because it was the first metabolomics research that had happened. It was a bit of a divergence from, I guess, what was largely immune-based. And for people back then who may not remember, but there was actually a bit of controversy around that time when I was doing the metabolomics around a particular virus that was tied to ME/CFS
Okay.
called XMRV, which was, I'm not sure if people know that history. It was kind of a bit of a dark time because it looked so promising and then failed so spectacularly. But I think there was a lot of research in that space towards immunology. And I guess there always kind of has been within ME/CFS. And I guess we see the same thing again with long COVID. We were just particularly interested in the metabolism aspect of it because of the symptomology that looked like it would be best explained from that. And, across the omics cascade, we have genomics, transcriptomics, proteomics, metabolomics. And metabolomics is closest to phenotype usually. So if there's any
Right.
biological thing that was going to look like closely represent, symptom fluctuation or symptoms at the moment. We thought that it was going to be potentially metabolomics. And we even did some investigation with some later studies which were highlighting particular symptoms within the cohort and their relationship to some of those metabolites as well. But I would say largely well received within the ME/CFS community, even in the metabolomics community, because it was well done, metabolomics research. I think the limitations probably outside of that, like I would still, go to a doctor's clinic and tell them what I do, and often I would get, doctors not believing that this was a real disease. So, I definitely. I think actually the main thing for me was this sense of fairness, for sure, that got me really interested, and it kind of made me want to prove that wrong, because you could see how unfair it was. And I knew, I could see just from what we were doing that this was something. So, I thought that was kind of an interesting time for sure.
I think that's something that we share there, and one of the reasons that we wanted particularly to publish a lot of the work that we've been doing in the genomic analysis of ME/CFS and long COVID was to establish unequivocally that there is a biological basis for these diseases, and that they should be on exactly the same footing as any other biophysiological disease. There's no hiding behind that. Simply because medicine at the moment doesn't have all of the solutions doesn't mean that we shouldn't be taking patients absolutely seriously and treating them with the respect and care that they're due. But I think we've still got a little way to go on that, but I think progress is being made. So tell me a little bit about some of the progression of that work. So I believe you collaborated with Stanford for a while, and you obviously know the great Ron Davis from that work.
So I probably should tell a little bit more. So that's where I did my PhD in the University of Melbourne, and so I also did. So I started a PhD. Which went for quite a few years, and I ended up finishing that up while I was waiting for some publications in around 2000 and at the start of 2018. And then I was pretty much in the university for a year, just doing some part-time work because it was hard to get funding within the ME/CFS space. And I was offered a role to work with Open Medicine Foundation in the US. And so that was kind of like a lifeline, because I was struggling to get funding for sure. And they offered a really, excellent job, which was to work as a science liaison and communicate across, the different centers that they had, but also look to chat with all the different types of researchers out there that were talking to Open Medicine Foundation, because they had a lot of people coming through, and they weren't, they didn't have a resource to get back to people or kind of chat to people about the research that they were doing and what they were interested in, and the different paths that could be followed, And so Linda was particularly interested in all the different pathways that, you never know what could lead to what. And, so Linda was instrumental in kind of maintaining me within this field for sure. Otherwise, I don't think I would probably not be here, because of just the lack of funding resource and the fact that I was effectively leading ME/CFS as a PhD student in my group. Like, no one else did ME/CFS. No one else was doing metabolomics in my group. I was kind of just like this one individual that would have just probably gone on to something else if I couldn't have got funding. Obviously, I was still always interested. If I could continue to have got funding, I probably would have dabbled, but not to be in the position that I am now. And so I went over and worked with the Open Medicine Foundation, which was, a wonderful experience to kind of understand how the foundation runs, and even just seeing from my part, talking to all different researchers across the world, and how the different types of research and ideas people had, and talking with the directors at the time, within the different centres at the Open Medicine Foundation who are being supported by Open Medicine Foundation, I should say, and getting them to connect with each other. So that was an interesting role for sure. And I think as part of that, the idea was that I could continue my research part-time at Stanford University under the supervision of Ron Davis as a visiting scholar. And so we sorted that out. And so then I was working at Stanford effectively, and part-time at Stanford and with Open Medicine Foundation. Unfortunately, the pandemic hit. And so that kind of put a dampener on what was really possible. It was especially hard hit there around the San Jose area and around Stanford especially. They really all locked down. I think they were the first places to lock down really within the world. And so from that, during that year, I decided to apply for a lot of grants throughout what I could get within the US and also within Australia. And we managed to get a few in discussions. So transferred, effectively managed to get funding to get back into Melbourne and had a large NHMRC grant and a couple of other grants as well that was able to help us set up a group again. But that was the short run of how everything went. But effectively at Stanford itself, I learned a lot of things from Ron Davis, I think here, especially about, managing and running a lab.
There's a recurring theme throughout this, which is fighting for funding to do work in the space. We'll come back to that because you seem to have fingers in a lot of different pies. But talk to me a little bit about your particular areas of interest here. You've been looking at metabolomics, metabolic profiling, and metabolite GWAS, and you're essentially, I think the hypothesis is that there are genetic perturbations or other reasons why energy production switches from its usual carbohydrate and fat sources and starts increasingly to use amino acids as an energy source, which is both more inefficient but also creates a burden of nitrogen that has to be excreted, and that can lead to buildup of toxic products like ammonia, for example. How has that theory developed, and how's it supported by the evidence that you've seen over the years?
The origin of that theory, and it kind of came together while I was working part-time at Stanford, and the origin of that came from my original metabolomics work. So obviously in Melbourne, even that first metabolomics work, we highlight that these amino acids are being utilized for energy through this pathway. That's what we saw, and that's what it looked most closely to. How I managed to find that was looking at the metabolic profiles. I actually looked through all different types of diseases to see which ones actually closely aligned with this phenomena. And one of the things that explained it really well was starvation and sepsis, combinations of things, which were like these acute stress biology components. And so from that, I was able to kind of look and identify, that amino acids and their usage is actually very, very important within these illnesses. And so what happens, I guess, during the body under stressful conditions is that you end up having amino acids being utilized for energy production, but also fats. But in particular instances, you can burn through amino acids much more fast or much more rapidly than lipids. And amino acids are actually very carbon saving. So I remember at the time Navial had published the metabolic features within ME/CFS, which is about 2017 or 16. And in that one, he proposed this idea of hypermetabolism in ME/CFS. And what he actually meant by that was when you found significant differences, they all seemed to be significant reductions in metabolites in ME/CFS compared to control, right? And that was just, but when you actually, what I noticed, and I noticed that was in our data as well. So I thought that was an interesting finding. But then what I noticed is there was reductions in the small metabolites. So metabolites are made of carbons and nitrogens and hydrogens, and they all have a certain mass, like, 1, 12, carbon, 16, so oxygen. And so when you actually count up the molecular mass, the scale, the metabolites that were small were actually the ones that were reduced, and the ones that were bigger were actually elevated.
Oh, interesting.
So what I looked at was, instead of looking at metabolites within NMR, because NMR literally counts the atoms.
Yeah.
I looked at the amount of carbon within the metabolic pool, hydrogen and oxygen. And even though we had a lot of reduced metabolites in ME/CFS, when I actually looked at the amount of atoms, the carbon, hydrogen, and oxygen were exactly the same between patients and controls. And I'm talking like fold change of 1.01, like very, very similar within that realm. But nitrogen was actually 10% less. So there was 10% less nitrogen in the patient's biofluids. So that's when it occurred to me that this lower amount of nitrogen might actually be quite important. And if the nitrogen's not in the metabolic pool, then maybe it's in nitric oxide or nitrates or ammonia or other forms, because we did measure urea. So that's not actually, even though we measure urea, that wasn't in that form. So that's what got me thinking about, well, could they be burning through amino acids more rapidly because of this? And then follow-up cell experiments from different labs and even our own work have shown that same thing. That's what's happening. So you get upregulation of the enzymes degrading amino acids, and even in the media, when you feed them, we get faster drops of amino acids from that media through the cells burning through that media itself. So amino acids definitely seem to be being utilized more for energy production, which isn't exactly abnormal, because stress cells will do that. It's, I guess, the level at which that's happening that is the concern. And the reason why it's really relevant is because, as I said before, amino acids are actually carbon saving. So what that means is glucose and fats both are burnt and produce quite a bit of, the amount of ATP to carbon dioxide that is produced, and that's the carbon dioxide you breathe out, is much more through burning per ATP molecule produced through fats and sugar than it is through amino acids. It's not much more, it's just a little bit more.
So ATP is the molecule that the body's cell use to convey that energy and to use that. So it's a bit like producing petrol from oil, I guess. So maybe a crap analogy, but
it's like I think what you're describing is probably a good way of putting it. The carbon itself, the hydrogen, the oxygen, they are producing carbon dioxide, which we breathe out, and the CO2 component, and amino acids produce less CO2 when you're burning it. But the outcome is that they produce ammonia or other toxic compounds. Now, your body has this thing called the urea cycle, where it's able to produce urea, which is a safe way of removing nitrogen from the body. And so that's the safe way of doing it. But if you that only goes at a certain speed. So if you are requiring amino acids in a short period of time to be burned for energy, you are inevitably going to burn more and make more ammonia and nitrates and all that sort of stuff. And that was a part of the basis of the hypothesis around this. Yeah.
So the fact that you didn't see the signal in urea is just because of a limit. It can only process a certain amount, and there's an excess on top of that then needs to go somewhere else, and that's creating these toxic byproducts.
Exactly. And I wondered, if I just looked at the most simple explanation, could amino acids being burnt more for energy cause the symptoms of ME/CFS? And I genuinely think they could, because it's not just if it's chronically happening, it's not just the production of byproducts, but if you think about some of the things that align with ME/CFS, like reduced peripheral blood flow. Now, one of the main reasons, blood flow isn't just about providing substrates and oxygen. It's actually a lot of the value is actually removing toxic byproducts from cells to get back to the liver and back to the kidneys to excrete. When you have reduced peripheral blood flow, you have reduced capacity to remove these compounds from different regions. But there's also the fact that amino acids play roles in other ways. And some of the areas that are first to get degraded or first to reduce in usage are things that the body kind of might see as less useful. So digestive enzymes. Like when you started using amino acids, one of the first things that stops getting made is digestive enzymes. And so that leaves your gut less able to break down dietary foods fast.
That's kind of akin to one of those real stress responses. The body is focusing on its core function just to keep the body alive, and it doesn't have room to process food or other central kind of activities that we would expect it to be doing.
Exactly, and I think that's one key part of it. And the digestive enzymes, you have bacteria in your gut that can do the job of breaking down food without digestive enzymes. But what happens is you start to get food going deeper into your gut and feeding different bacteria that can grow in different environments. And that's when you start to get more pathogenic bacterial varieties, potentially, which are often associated with either eating too much or some of these chronic stress problems. But then also glutamate and amino acids have many other roles within the body. So glutamate's a key neurotransmitter. They're really important for making nucleotide bases, which is the base of ATP, which is for energy. So, but also all your gene blocks and different genetic structures, they all require amino acids and nitrogens to be added on to these compounds to actually make them. And another one is another really interesting one is collagen turnover, because collagen, like lipid membranes, collagen's made of amino acids and it's constantly cycling. So instead of repairing different components that get damaged within amino acids, it will just cycle out. So you'll just get collagen being degraded and then remade and all sort of stuff. But while it's in that pool and breaking down, it's making amino acids, which can then be thieved away for energy production.
Do you think that's a connection over to some of the hypermobility, the EDS type of disorders that we often see comorbid with ME/CFS?
That's what I'm thinking. I'm wondering whether that collagen degradation and poor reforming might be part of that process where you have amino acids being ongoingly utilized for energy production and that reservoir being taxed effectively because the patients are more relying upon this within certain cell types. And look, the cell types that we use are often immune cells, so it may not represent all cell types. That's why it's hard to know. We haven't looked at every cell type of what they're doing biologically. So it's definitely, it can't be across the board that this is happening in every cell, but the biofluid, like the cell-based experiments, but also biofluids like blood is what represented some of these changes for us early on. And so that was the idea behind the nitrogen. We call it the nitrogen hypothesis because it's more than, even though people get very fixated on the ammonia component, but it's actually a lot more about, because I think people think, oh, ammonia, oven cleaner, or, that is obviously quite toxic in the brain. And certainly is, and certainly could have a role in it. But there is, a lot of other components to this. The body will probably not designed to utilize amino acids ongoingly. That's not an ideal situation because of the negative consequences. So if it's carbon saving, so if amino acids were good for you and didn't have these negative byproducts, you'd be using them all the time. But because they do, you definitely don't. More like a backup, like a diesel generator is what we usually use as an example, as a backup to grid power.
So, I mean, it's fascinating though, because The hypothesis supports one of the key observations about ME/CFS and long COVID as well is just the sheer number of symptoms and the variety of those symptoms across different organs through the body. And I think that's one of the reasons why clinicians have found it so difficult to get a handle on. It's probably one of the reasons why pharma companies have found it so challenging a disease to study. In many ways, it's an ideal disease to study for them because you have tens of millions of sufferers around the world, and there is no competition out there of effective therapies. It's something that in principle they should be interested in working on, but the level of activity is much lower than it's been in many other diseases. Have you found traction with those sorts of organizations, or how is the hypothesis received?
I think in some contexts, people are interested. Definitely people, I presented this to a few different groups. People like Paul Berg, for example, who is a Nobel Prize laureate in biochemistry, love the idea. It's an interesting idea. And when I say it as a hypothesis, I call it the hypothesis is really just about the biology underpinning a broad array of symptomatology as a problem that might be consistent across patients. But I do think there's genetic differences and heterogeneity, which there are many different reasons why this might be happening. And I think that's the key part. There's many different reasons why this may be occurring for genetic different reasons. And I think that's the key difference of where this lies. I'm not trying to say this is a biological reason for the disease, other than to say that this seems to be to me a common funnel to which we get similar expression of symptoms. But in terms of pharma, I've chatted to pharma a little bit. The real problem with ME/CFS over the years that I've been researching it, I've known of a number of trials that never even got published, like companies who had trials things within ME/CFS, or maybe even some supplement companies, that have trial things in ME/CFS that just always had very high placebo problems. So the group has a high placebo, and then from their perspective, the treatment does not surpass that placebo, and when they do a proper trial of it. So I think the outcome measures is the main reason large bias companies don't touch it. I've chatted to a few, and that's the reason. They don't have an objective outcome measure that they would be really comfortable with, and they know that there's a history of trials failing within this illness, given with its being subjective markers. Yeah.
So this kind of brings us on to things that we're looking at working on together. You've been a proponent of precision medicine around ME/CFS, long COVID, and other areas. And we've been investigating the opportunity for using existing drugs that are out there that are safe and well tolerated. We know how to. Well, we can obtain them. We know how to dose them. What do you think the possibilities are for drug repurposing? Is this the way forward for these diseases?
So that's something that I'm quite interested in. I think the drug repurposing has quite a bit of potential. Obviously, there's a lot of medications that are already available and have gone through safety testing. If you don't go off-label drugs, if we were to design something specifically, like we'd have to find the mechanism of ME/CFS, or a mechanism within a subgroup, identify a drug that fits that particular subgroup or fits that mechanism, and then go through all the safety testing, and it can take a long time. So to me, the off-label, because it's faster and we have so many available drugs that have gone through safety, it just makes sense of working out fits. But that's not the only reason. One of the big reasons is a lot of ME/CFS patients, I've known quite a lot of ME/CFS patients who have actually had considerable improvement on certain medications. Now, it's never the same medication, but there's something about certain meds for certain people that does seem to hit, and some people just occasionally hit on a winner. And it's not very well understood why that happens. But to me, based on what we see in the biology, I think it could make sense that you can, if there is a chronic stressor creating, and you have a genetic susceptibility for this biological manifestation of ME/CFS, you can alleviate that chronic stressor in a meaningful way. Then it does make sense that a few people can, improve dramatically. And it's like, well, it's not everybody, but maybe it's a way in which we can eventually kind of understand each individual. And so I'm very interested in the precision medicine approach that your company represents, and how we can potentially test to see, how this effectiveness, and how precise it is, and what we're able to do with it, I think is obviously transformational, and definitely something that needs to be done as a high priority, in my opinion.
I think one of the things that's always encouraged and frustrated me at the same time, you have studies like the observational Treat Me study that OMF ran, basically asking ME/CFS and Long COVID patients which therapies they'd tried, or which supplements, which lifestyle changes they had tried, and just asked them for their impression of what was working for them. And I think what is the frustrating bit about that is that nothing works for everyone. There is no silver bullet that's suddenly going to fix the disease for everybody. But there are consistent reports from hundreds of patients in many cases that 20-30% of patients felt that a particular therapy was helpful to them. Maybe in a little bit, but maybe in a much bigger way. The challenge always is, as you say, in clinical trials, that isn't a big enough signal necessarily, particularly if you have a lot of placebo effects. You have either patients who don't benefit, or you have patients who are recording benefit while they're actually on the placebo medication. And this, this seems to be one of the fundamental challenges of dealing with a very, very heterogeneous disease, a disease that potentially has lots of different things going on at different times. It may mean that a drug doesn't work for that patient at that particular time because they're not in that stage or that form of the disease. So being able to find a way of pre-stratifying those patients into ones that you have a really good genetic hypothesis for why they should be responding to this medication, and then enriching that trial population using those kind of tools seems to me, at least, to represent a slightly different and a way that is more likely to yield results than just doing a blanket clinical trial across everybody who has an ME/CFS diagnosis.
Effectively, we're trialing people, and the way we've been trialing people is to select people who happen to meet an ME/CFS diagnosis and meet that towards a particular medication. And all you're arguing here is, rather than a diagnosis based on a battery of symptoms and those definitions that meet a diagnosis, and you're talking about an individual who's met that being, and at different stages of disease and all different types of factors going into a trial. Instead of doing that, you're basing it on a genetic subgroup within that cohort. So it's not just that they have ME/CFS, but they also have to have these genetic features to then be put through into a trial. So almost like a subtype of this. And I think that, that definitely has validity and very well could be, at least by bringing in that biological differentiator. I can't say whether it's going to be more likely, but theoretically, it should be more likely. Theoretically, it should be more likely to definitely to have improvement through this space. And the only way I say that it, I can't say for sure is because the idea is to test it. This is why it's so important to actually go out there and do these trials to see whether this is actually doing that, and trialing that platform itself. Because if you are in the right area and this is actually hit, the massive impact that you can make in such a short time is just so tremendously large that to me it's just so critical to look for that, for sure.
And you used a phrase that I really like, which is that we're trialing people here. And this is one of the things that's probably not so immediately obvious to listeners. You sort of alluded to it a little bit earlier about how long it would take pharma companies to actually bring a new medicine to market. On average, that's, let's say, 12 years. We can argue, whether AI has made a difference to that or not, but historically it's 10 to 12 years. But that's actually in a fairly idealized scenario where we have the tools, where we have tests or assays, where we have, unfortunately, animal models that we have to use, which simulate the disease. And I think it's fair to say we really don't have many of those tools in ME/CFS and Long COVID. There are no good models out there that we can rely on. So, in principle, if a new medicine were discovered today, it's going to be at least 10 to 12 years before it comes to market through that traditional kind of approach.
And the only way they could discover a new medicine is if they worked out the mechanism. So they worked out a group of people diagnosed with having ongoing fatigue, post-exertional malaise, brain fog, unrefreshing sleep for over 6 months, typically years, most likely, and whatever other array of symptoms that they have, that particular group of people defined by this condition or this diagnosis are then going to have all the same biological mechanism, when them themselves have fluctuated so much within their own disease, in trajectory, being worse and getting slightly better, elevating baseline up and down all the way through their journey. But at one time point, they're going to be within this all similar mechanism. To me, it just feels that seems very, very unlikely that there is a common biological mechanism that explains or that is causing ME/CFS to the point that one drug is going to be a silver bullet. I feel like that is extremely unlikely in my opinion. You could get a silver bullet treatment that's going to help everyone by 10, 15% or alleviate a whole bunch of people. I could see potentially that, but really making a substantial improvement. People, people was an intentional term, and that's why you picked up on it, because it's generally people, we're all we're genetically different. We have genetic differences between us. And our capacity to respond to stresses differs because of these genetic features that we have. And you could go your entire life without realizing you have genetic abnormalities until you reach a chronic stressor and how your body responds to that, to the severity of what may be an ongoing severe, acute infection. And then whatever happens because of that can kind of undo and make something like ME/CFS. And I think, when you speak about ME/CFS to mitochondrial disease experts, they think you're just describing mitochondrial disease. That's, it seems like it's the same thing to them, which I think gives you a clue as to what we're really looking at.
Yeah, there's an interesting sort of implication of what you're saying there, which is, it's not just that one drug may not help everybody. It may be that we need more than one drug, even within an individual patient, to address multiple of the symptoms that they're showing. Obviously, we have to run. We have to walk before we can run. But do you think combination therapies have a role in ME/CFS?
So I do think combination therapies are going to be great for ME/CFS because of some of the combinations of symptoms that they have. But like one example would be the gut issues that patients have. If they have medications, because they often have impacts on the gut and negative side effects within the gut, if they can exacerbate the gut issue, they can make patients feel worse. And if a patient can probably feel that they're getting worse, they're probably going to be worried about long-term severe outcome and worsening of ME/CFS, which is a major fear for a lot of patients. So them wanting to stay on that medication is not going to be something that probably is ethical or stress, and I think combinations of medication with that, definitely. But I think one of the really important things with medications is not just the combinations, but the dosage and how you kind of start low and ramp up, and how the way in which patients can actually begin on these medications is particularly relevant. And even on different surveys where people talk about drugs that they've tried or anything like that, or if they talk about medications, the dosage is really relevant. Like, how much did they try? What did they try? Like, what was the max dose? How did they start? Did they ramp up, or did they just put on to, a major dosage? We had like a lot of feedback from that. We had a paper around metformin and its potential value. And we were thinking about a low dose version, but we got a lot of feedback from people on, the full dose of metformin that had massive levels of gut side effects. And, that was a really interesting experience because, that really is critical, that low dosage. And there really isn't very good regulations about what those dosages are meant to be. And so when we're using off-label drugs, that dosage level becomes really relevant.
We know, for example, in the context of GLP-1s, that is potentially an issue. We have a very frail patient population, and there are reasonably significant side effects associated with the full dose when it's used for obesity and diabetes control as well. So, yeah, that's very. We have a very frail patient population that we're dealing with in many ways, a very sensitive patient population. So being super careful about dosing in the context of a trial in order that you see the positive effects you want, but don't see so many adverse events that the drug gets ruled out, I think is absolutely critical.
Yeah, I think I've noticed that a few groups doing trials more recently have done a good job of this. I think they've been considerate with their approach and how they do this. And I think that's really important. But the backlog of data that we have on patients who have trial medications may be turned off a medication because they started on it too high.
This has been a really fascinating insight into the thought process behind a lot of the research that's going on. You've been in the space for about 15 years now, at least. How do you feel broadly about the profile of ME/CFS and the impact that its research has had, or you have realistic expectations that it will have in the near future? What's changed over that time?
So I think what's changed is a greater emphasis on PEM and a greater emphasis on individual symptoms, a greater inference. I think most of the research, even up until a few years ago, was largely based on this idea of everyone having the same biological mechanism underpinning. And so I think I'm seeing a lot larger switch towards people accepting the idea of a heterogeneous population because, I guess, of the way it's diagnosed and because of the biological outcomes, which we haven't been able to nail down a mechanism. So,
and we're seeing the genetics that supports that now,
and the genetics that supports that. And I think our data supported that. All the biological data supports.
Yeah.
You see subgroups, you see some clear phenotypes that are quite different within cohorts. So I think that's been a really critical one. Obviously, without ever finding the mechanism, it's hard to feel like you've made progress, because you've been here like trying to understand what in the field for 15 years, and you're like, well, what has happened, because that's obviously the landmark to which we're trying to get to. And so until that's nailed, it's really hard to kind of go through the rest of the process, and for people listening at home or people listening to this, most of the biological research, pretty much everyone that I interact with within my university in other diseases, all that research is based on understanding the mechanism first. That's not even a question for majority of disease. Like you understand the mechanism, that's when you can develop diagnostics. You go, well, this is a measure of the direct mechanism. Oh, it costs tens of thousands of dollars to look at this. Let's find a small diagnostic that kind of mirrors it, in a trial, and we can find that in the blood or the urine that's representative of this disease and this mechanism. So that's like the field of diagnostics, working on simplifying a marker so you don't have to spend so much money to identify this in cohorts, and then you can screen it in pathology.
Yeah.
Well, you understand the mechanism, you develop a drug that targets it directly. There's people working around the clock looking at mechanism, like disease drugs that can target the mechanism in different ways. People who take that mechanism, put it into a model or into a cell, or understand it in the lab, break it apart into different pieces and try and understand all the aspects of it. This is the entire fields of biochemistry and biological researchers in disease, often based on diseases where the mechanism is already found. And so it is frustrating, for sure. And I guess it's frustrating for everybody involved in this field, absolutely. But I think, but I do see progress in the way that we're thinking about the problem, because I think that's, part of the reason the mechanism is being so hard to find is because it's not one mechanism. And that to me is a really important transformational shift within the field.
I mean, I haven't been in the field for anything like the length of time that you have, but I think that realization and beginning the acceptance that it's a heterogeneous disease with multiple intersecting mechanisms that have impacts throughout the body in different ways. Exactly the kinds of hypotheses that you're generating and creating evidence for. I think those, instead of just having a blank landscape that we really don't know what to do with, they give us areas to go and focus on. And I think, I'm in the five or so years we've been working in this space, I think that's what we've observed. Some of the hypotheses are starting to have good evidence behind them, and as you say, that gives us a place to go look for new drugs. So let's end on a hopefully hopeful note. Where do you think we're going to be in? Well, I'm not going to ask you to talk about 15 years because I think none of us would know. But where do you think we're going to be in 5 years' time with ME, CFS and Long COVID? Do you think we're accelerating our progress, or is it going to take a while longer?
I think we will be more progressed than we are now. Progression is happening. It's just hard to be visible in terms of the process with when a mechanism is an ultimate goal. But we have so many different groups thinking about the heterogeneous problem. Obviously, your company, but there are a lot of different groups thinking about this problem. Our groups, we have our own different technologies and things that we're working on, and different groups around the world that I know of. So I think that, but also the advent of AI is actually going to be very useful for dealing with a complicated illness and heterogeneity within that, understanding individuals over time and modeling for that. I think within 5 years, we could really see a lot of really useful tools being developed for patients to manage their disease. And I think it's possible that we do have subsets linked to medications within that 5 years as well. That's what I think. Well, actually, that's what I would say is more likely to happen within the next 5 years, that we will have certain subgroups of ME/CFS linked by a biological marker to represent them as a group that's going to benefit from a certain treatment. So
Yeah.
that is my thought for the next 5 years. So I think it's going to be really, really, really great. That would be my conservative opinion. It could be far better than that, but that's my conservative thought on what will happen over 5 years.
No, I must admit it's a hope and an expectation that I share. I think.
Yeah, that's an expectation for me too.
Yeah. What's less obvious to people outside the space is, we have probably 20,000 active drug molecules that have been tested for one thing or another, and one of the side effects, if you like, of finding lots of genetic and mechanistic associations with the disease is that many of those have been encountered in other diseases. And there may very well be active molecules that were tested in fibromyalgia or, in asthma or some other disorder, that end up actually being useful to patients, and for which we may have a very strong biological hypothesis, and in fact may have evidence for. And I think I'm very hopeful that groups like yourself, working with clinical populations, are going to enable us to do the trials that prove the connection between that biology, that compound, and a subset of patients for whom that therapy is going to be beneficial. So I think reasons to be hopeful. And if I may, I think I was talking to Sonia Chowdhury of Action for ME recently, and we were talking about the size of the prize here. And I think that's something that gets overlooked when we talk about grant funding. These are diseases that account for pushing 1% of GDP in major economies when we're talking about the impact they have on patient lives and their ability to be active, socially and economically. And that is a productivity gap that politicians really should be interested in closing, because if we're this close with a number of, well-designed, relatively straightforward, relatively cheap clinical trials between us and realization of that benefit, that is not the hardest gap in the world to close.
No, I think that's an extremely important, valid point. And I think, we'll be thinking about along that lines. And I think politicians would, and they are interested in this. They are. I think that's just giving them the ammunition to be able to make better cases. And sometimes I think, numbers on the economic impact and some of the things like modeling biological outcome, like modeling prediction of numbers and that GDP loss. And these are all very relevant. I think becoming more relevant as people become smarter about how they quantify disease, especially its impact on function, especially its impact on people being able to work. It's not something that I think it's something that's being maybe disregarded because they just haven't had the numbers or the understanding of how that kind of adds up. And it adds up, as you say, to a massive, massive level.
Well, also, your hope for 5 years that we actually have successful medicines that are working for subsets of patients, that's one political cycle. So we're now in a point where we could actually see success within, measurable within a term, which I think is it's one of those actually important points in terms of getting support for this. People would love to be part of the solution for this patient population. And I think there's every expectation that we're close enough now that can actually be the case.
Yeah, I think so, definitely. I think there's actually really strong cases to make for this. And, I think if we make that case, I could see some benefits coming. And I think, we've seen that in Germany recently.
Mm-hmm.
We've seen that in Netherlands. I think we've seen that in a couple countries where governments have listened and seen those numbers and seen that potential and gone, that is actually a big deal. That's something that we could make quite a large difference, in that population. And so that's something that we could be all hoping to see from our governments, wherever we respectively may be.
Fantastic. Well, that's a hopeful note scientifically and for the field going forward. So I just want to say thank you, Chris. I know I know we're interrupting bath time for the kids. So thank you for finding time on your vacation to speak with us.
No, not a problem at all. Thank you very much for having me on, and it was a pleasure to be here. Thank you so much for putting this on and the work that you're doing within Precision Life, and we'll continue to work together and hopefully come up with something that is transformational.
We're really looking forward to it. Thanks very much, Chris. Much appreciated. If this episode changed the way you think about precision medicine and chronic disease, share it with someone who should be part of the conversation. For more discussions on how a deeper understanding of disease biology is reshaping healthcare, follow Biology Matters wherever you listen. You can also learn more about Precision Life and the work that we're doing at precisionlife.com. And remember, the better we understand disease, the better we can predict, treat, and prevent it. Biology Matters. Until next time.